Good morning, everyone. So my name is Richard Forsgård. It's nice to be here to present my sort of research career, my work so far. I spent the last 4.5 years in Sweden at Örebro University, where I worked as a postdoc researcher. But since last September, I've been living in Finland again, and I'm now currently working as a grant writer at Örebro University of Applied Sciences. So I'm no longer involved sort of in day-to-day research, but I'm happy to put on my researcher hat today and tell you a little bit about the intestinal barrier, which is a topic that I spent the last 10 years or so researching. So let's start with this. This is probably a familiar picture from high school. So this is the intestinal tract. We eat food, we absorb, digest the necessary nutrients, and then we poop out the rest. It's an actually very efficient system. We only, like the average healthy person that we lose only about 9 grams of carbohydrates, 6 grams of protein, 4 grams of fats in our feces. All the rest we take up. And it also handles a huge amount of liquid every day, about 9 liters, and we only lose about 100 milliliters of that. So it's a very, very efficient system. It was evolved to do, and it does it very well. So I'm not going to go into the details of the absorption mechanisms, how we do that, but for the purpose of this presentation, I think it's important to know that there are certain anatomical structure that make this absorption process very efficient. So if we would think that the small intestine, where most of the nutrient absorption happens, would be just a smooth tube, a smooth cylinder, the surface area would be that of a pillowcase, roughly. But that's not the case. The small intestine actually forms these types of finger-like villi, which then hugely increase the surface area, and the actual surface area is somewhere around half a badminton court. So we have these sort of mechanisms that makes this system very efficient. Well, of course, we have an efficient system that's very good at, it's a large system, it's very good at taking up stuff from the outside. There's one obvious drawback, and that is that there are lots of bad stuff that also wants to come in. So it needs to balance, it's a balancing act. So we need to balance this absorption processes with barrier function, which is the GI tract's second sort of main task, that it needs to keep out all the bad stuff that we don't want, that could possibly make us sick or unhealthy otherwise. So how does it do that? I'm going to introduce that so that we build the intestinal barrier here together. I have a daughter who's very into Legos, so it's a pretty similar process. So we start with the enterocyte. That is sort of the basic building block of the intestinal barrier. It's responsible for nutrient uptake. We see that it has all the necessary proteins, all the necessary mechanisms to take up nutrients very, very effectively, very specifically. So there are specific transporters for sugars, for amino acids, and that sort of things. It also, the cell membrane that all cells have, the enterocyte cell membrane, it forms a physical barrier between the outside and us. So let's stack these cells together. We can make sort of the villus structure here. We have these. Of course, that's not enough. We somehow need to link these cells together. Otherwise, it would be quite a leaky system. So here we see the white is the outside, the lumen, the intestinal lumen, and then here it's not really showing here, but that's sort of the inside, the blood circulation side. So we need to link these cells together. there are many different types of cell and cell connections, but in the context of the intestinal barrier, tight junctions are probably the most relevant one, the most important one. So these are protein complexes that then seal the space between adjacent cells. So now our sort of barrier is also sealed between the cells, but of course we need something more. Cells do not live forever, so we need we need a supply source for these cells. Enterocyte, their lifespan is about 3 to 5 days, so there's a very rapid turnover of cells going on all the time. So our sort of intestinal mucosa we have a new intestinal mucosa every 5 days. So what we need are intestinal stem cells. These stem cells reside here at the bottom of these villi. They're called crypts. And stem cells, they are cell types that can differentiate into other cell types, and intestinal stem cells can then differentiate into other intestinal cell types. So let's put those there at the bottom. Here they start differentiating, and then they make their way up of the villi, and then they die, and we lose them in pieces. Okay, now we have those. We have a supply source, but they look a bit lonely. They need some help. They need some support. We have Punnett cells there with the stem cells. These are specific cells that then support and regulate these intestinal stem cells and their differentiation process. But they're also very good at sensing microbial products, and then they respond by secreting different types of antimicrobial peptides. There's some speculation that this is because that they're like protecting the stem cells there, so it's important to keep microbes away from this stem cell niche or stem cell environment. So let's add those there as well. Looking good, but maybe we could be a bit more proactive in our defense. So let's add goblet cells. These are cells that secrete mucin proteins, which form our mucus layer that sort of lines our intestinal epithelium, the enterocytes. And the mucus layer is very good at keeping bacteria directly away from the epithelium. So this mucus layer is a very thick sort of mesh-like net that traps intestinal bacteria and keeps them away from the epithelium. So let's add those there as well. Yeah, it would be nice to maybe get some information what is going on in the lumen. So we have this sort of enteroendocrine cells. This is a sort of umbrella term for several distinct cell types, which produce different hormones and peptides that control different metabolic adaptation to for example, to nutrients. One of these hormones that's very topical now is the glucagon-like peptide 1. That's the Ozempic weight loss drug, mimics this protein, this peptide, and then suppresses appetite, and it sets some other metabolic functions as well. These also connect. We have a huge nervous system in the intestine. It's called the enteric nervous system, sometimes referred to as the second brain. These cells connect to this enteric nervous system. They secrete these enterochromaffin cells, which is a type of enteroendocrine cell. It secretes serotonin, which you probably know in its effects on mood and how that works with depression, for example. But the intestine contains about 90% of all body's serotonin. And it stimulates the enteric nervous system, producing sort of intestinal movements there. But now, recently, researchers have also found that these enteroendocrine cells can actually directly synapse with these enteric nerves, which is a very interesting concept because that gives us a very because the enteric nervous system then connects to the vagal nerve, which connects to the brain, which gives us the sort of gut-brain axis. So what this means that these cells can then synapse directly with the ENS means that we actually get we have a rapid sort of information highway coming straight from the lumen into the brain. And there's some interesting findings on this sector. So, for example, in mice, it has been shown that these cells can differentiate between natural sugars and artificial sweeteners. So even though, and they make the mice prefer the natural sugar. So even though the mice cannot taste the difference, it still goes to the natural sugar bottle and drinks from that because the brains or these cells tell it to that this is the good stuff and this is just artificial sweetener. So yeah, I guess there's some truth to trusting your gut in certain situations, but yeah, there's not so much human data on this yet. Yeah, let's add those. Now we can also add some bit of the enteric nerves there, how they are connecting to those. Then we have another type of sort of chemosensory cells. These are tooth cells. They're important against parasite infections, which is a very sort of relevant evolutionary mechanism. We have a lot of intestinal parasites, so we have some sort of cells that are specialized in handling those. And then finally, we have sort of M cells that are located on the epithelium, and then they take up microbial antigens from the lumen, and then they present them to the underlying immune cells. The intestine is a very active immunological tissue. There are a lot of different types of immune cells that are sort of monitoring what is going on in the lumen, what type of microbes we have there. Is it something that should elicit some sort of response or not? Is it sort of a good bacteria, bad bacteria, and so on? So it's constantly in sort of crosstalk with the intestinal lumen. And as I already sort of, no, it doesn't show that. But this is, of course, we have the intestinal microbes there, and the microbial products is a huge ecosystem that regulates this, is in constant crosstalk with this system, and sort of can support the barrier. Some microbial products do that they support this system. Then we have, of course, bad actors there that then can disrupt the system or and then make us sick or whatever. And so it's a very complex, dynamic system that's keeping us healthy, keeping all the bad actors out. But how can then substances cross this intestinal barrier? So there are basically two pathways. So they can go through the cell. This is called the transcellular pathway. This is very well regulated. Like I said, there's specific transport for different types of nutrients, different types of substances. But then also they can go between the cells. The tight junctions are not a perfect system. Sometimes they break up. We can have this paracellular pathway is very unspecific. So there can be sort of bacterial products going through. It's not so kind of well regulated as the transcellular pathway. So what we usually scientists, when they're talking about increased intestinal permeability, you see this in the news as leaky gut. So what that usually means is that there's an increase in this unspecific sort of paracellular pathway. But let's take a closer look. How does this then connects to disease and so on? It's some sort of pathophysical mechanisms. So let's say we have some sort of injury, some sort of disorder that causes that the barrier function is somehow disrupted. We have increased intestinal permeability. This means that there's an increased flux of harmful substances possibly going into our system. And like I said, it's a very immunologically active tissue. So then we have a lot of sort of immune activation going on because of this increased flux of this toxic or noxious substances. And then this feeds back to increased intestinal permeability. Unfortunately, we know that sort of inflammatory mediators, they also increase intestinal permeability. So they break down this barrier even more. So then you end up with this sort of vicious cycle that then drives possibly this disease. However, it's still very much debated which comes first. Is it the inflammation or the increase in intestinal permeability? There's data going both ways, so we don't know actually. But yeah, we know that increased intestinal permeability has been associated with several diseases. Inflammatory bowel disease, that's the Crohn's disease, ulcerative colitis, irritable bowel syndrome, IBS, celiac disease, maybe the gluten, the gliadin peptide in the gluten. It has increased access to the immune cells beneath the intestine, and then that drives the celiac disease. Some autoimmune disease, rheumatic arthritis, for example, some neurological diseases even. Again, the increased intestinal permeability then possibly drives sort of inflammation and then neuroinflammation and leads to neurological diseases. Then it's also involved in metabolic syndrome, type 2 diabetes. These are usually characterized with some sort of low-grade systemic inflammation that may arise from this increased intestinal permeability. But now, to sort of my final thing is to then, if we look at this in the context of human lifespan, so when we're born, the intestinal barrier, it's developing, it's learning which are the good bacteria, which are the bad bacteria. But as we age, aging affects all tissues, including the intestinal barrier. There's a concept of inflammaging, which means sort of that as we age, that drives systemic sort of low-grade chronic inflammation in different tissues. And there's some evidence that the intestinal barrier actually could be driving some of this process. And that's very much, the inflammaging is very much connected to sort of poor health and frailty in elderly and even sort of mortality. So if you less inflammation means that you're probably living longer. And centenarians, people who live over 100 years, their sort of gut microbiota resembles much younger people. So there's some sort of crosstalk there that also drives the aging process and the detrimental effects that come with that. So then we can go from healthy to disease. Maybe we have some sort of genetic predisposition that already we get some sort of chronic disease even at an early stage, or then it can happen later. But of course, we do not age in a vacuum, where the term exposome refers to all the stuff that we're exposed to during our lifespan. So we have a lot of data sort of showing that, of course, diet is very important for the intestinal health. Mediterranean diet, for example, has some anti-inflammatory properties that probably help also the intestinal barrier. We are taking, some of you may eat probiotics. Those can be helpful in some cases. Dietary fiber is very important for gut health because the intestinal dietary fiber sort of supports the growth of the good bacteria in the intestine, and then we're exposed to different sort of chemicals. There's some data showing that these perfluoroalkyl substances, these forever chemicals that are in our environment, are increased in people who have IBD, for example. So whether that's a cause of the disease or some other mechanisms, we don't know yet. Stress, so work-life balance, how stressed we are. We all know the nervous shits that some we will get, some more than some others. So that's also, stress is a very important factor for gastrointestinal health. We have data showing that even early childhood sort of trauma or stress can then show up later as some sort of, for example, IBS or IBD. And stress often exacerbates IBD symptoms and so on. We are constantly learning more about multi- and transgenerational effects, some sort of changes in not in DNA, but the stuff that regulates DNA, how our genes are read and how our proteins produced from those and what genes are active. Those can be also hereditary. We are constantly learning more. It's quite a new subject still, but we're still learning. So there probably is some how our parents' lifestyles possibly affects us also. And then a new term also coming from the recent years is chrononutrition. So it's not only what we eat, it's also that when we eat it plays a factor on that. We know that shift workers, for example, are more susceptible to metabolic disorders and so on. So I'm going to leave you with this. Take care of your gut. And that was all. Thank you.
Thank you very much for this very fascinating presentation. As a historian, I was thinking that we think that the world is complex, but now we can think that we are inside very, very complex. But now there's time for a couple of questions. Please.
Hi. So you mentioned the stem cells. So are they producing all those cell types that you mentioned, or is it only like?
Yes.
Yes. Okay. Yeah.
So they sort of differentiate. I don't know the actual how it's regulated. Which type.
Okay.
I haven't looked into that so detail.
All right. Thank you.
Okay. There's one question from Velma, please.
Yeah. Hi. I couldn't resist the chance to ask a bit about something that I spent a lot of time working on, and it's interesting to get maybe an opinion from the different side of the puzzle. So I was studying Parkinson's disease and gut microbes for quite a while, now studying something else. But you brought up this possible connection of like barrier issues to neurological, neurodegenerative disease. Do you have any sort of your own opinion or take on this? Because it is definitely something that is still even in that field, a bit controversial. And how strong is this connection? And is it sort of, in your opinion, scientifically thinking, a plausible hypothesis that there is such a strong connection to neurological disease and the gut?
Yes, that's a good question. Regarding sort of Parkinson's, I think there's, the sort of hypothesis that it actually originates from the gut is very, very prevalent at the moment, and its connection to the gut microbiota, as you said, and as you know. But that's a good question. The problem here is that we have no idea what is normal permeability. So in every study, we're always comparing to a control situation, to a control group. But we don't have an actual sort of clinical measure that if you're above a certain limit, you have increased permeability. And I think that also sort of kind of restricts what type of conclusions we can say about a specific situation, specific person, if they're more susceptible to a disease, certain disease or not. I think there's some sort of, I think it's a very good hypothesis, and I'm in favor of that. I think that's certainly possible. We see, I know I had a colleague, and he's still working on this in Örebro, and he's trying to see this sort of the connections and differences between Parkinson people and IBS people. And there are some similarities in their sort of gut health and gut function. So there's definitely some sort of link here to also the gut. But like I said, it's still quite difficult to say because we don't have sort of reference value what is normal. Then we have a lot of different types of indirect measurements. How do we measure intestinal barrier function? And some of them are good, some of them are not. So sometimes you see a scientific paper that, in Parkinson's, permeability is increased, and then you look into the actual paper, they use some sort of one marker that is indirectly linked to barrier function, and then making this conclusion of that. So that's some of that sort of kind of murkies the waters a bit here. But I think definitely a subject worth looking into more and actually doing some comparisons between, like I said, for example, with Parkinson's disease patients and then some sort of maybe IBD, IBS, and see if there's some similarities.
Good, thank you. I'm happy to hear there is also sort of some conviction on the more human gut barrier focused side that this may be something worth pursuing. Thank you.
So maybe this is quite related to what Velma just asked, that to me as a non-expert, sometimes it seems that there's some kind of a hype around this sort of gut health, at least in sort of popular media and so on. So do you think that this is the case? And if so, is it just on sort of on the side of how scientific results are reported in the media and not necessarily in the studies themselves? Or is there also something hype kind of bubble going on in the studies themselves? And if so, why do you think that this might be the case?
Yes, you're absolutely right. There's a lot of like the gut-brain axis was a huge thing about 10, 15 years ago. There was a lot of stuff, still a lot of stuff going on, how the gut is connected to the brain and how the microbiota plays a role in that. We had some studies where they took sort of fecal matter from depressed people, put them in rats, and saw that the rats became depressed. So that's a very much a hypothesis-driving thing that, okay, this microbiome actually makes these people depressed. But I think the hype is a bit died down now, which I think it's sort of going back a bit sort of to where it should be. It's not so hyped because, to be honest, there's a lot of shit science on this area also, making very, very big conclusions out of, okay, we have thousands of microbes in our gut, and then we see that in certain population, 2 of them are bit down. Is that relevant? Maybe, maybe not, but then you see that there's a huge media hype around this very, maybe a negligible thing. So there's a lot of, we still don't know. There's a lot of hype, like you said, but I think it's going in the right direction now. But I sometimes do that. I don't know if somebody else does that. If you see a media headline, then go back to the press release and then go back to the actual article. It's a very exciting process to see how the message changes from these. So I highly recommend doing that. It's very much fun to see how the broken phone effect, how the message amplifies during this.
Thank you. I think it was a very good piece of advice to all of us, and concerning all science and all science information we get.